Welcome everyone. Thank you very much for attending. This is going to be a seminar talk for Quantum BC at the University of Victoria. My name is Thomas Baker, and I'll be emceeing this event. So before we get too far into the program, let me just say that we acknowledge and respect the Lekwungen, Songhees, and Esquimalt peoples on whose territory the university stands, and the Lekwungen and Saanich people whose historical relationships with the land continue to this day. So today we have a very special treat. Reuven Gordon, one of the there, now you get live captions too, in case you want a transcript. No, it's there's no, it's on the other screen. Okay, so this, there we go. Okay, there we go. All right, yeah, I think that's just the disclaimer to let you know. Okay, yeah. Recorded. So today we have a special treat. We have Professor Reuven Gordon, who's runs an integral research group here at the university, and is going to be talking today about isolating and coupling single photon emitters with nanoaperture optical tweezers. And I'll just say that among his many achievements that he's had over his career and recent overall success, that he is a member of the IEEE, the SPIE, and the Optica societies. This is quite an achievement to be so involved and so productive on things. So without further ado, here's Reuven Gordon, and I'll let him take it away.
Thanks, Thomas. I'm actually a fellow of those societies. Not just a member, but okay. Great. Thanks for having me to give this seminar. It's good to see so many people showed up, at least online. In this room, it's only Thomas and I because we're in a different building over here. Okay, so I want to talk about some of the quantum-related efforts that are going on in my group, and some of these will be related to the students that were funded by this Quantum Create program, which is a great program, and they've had a lot of success. So it's great to be able to highlight their successes so far. Okay, so to motivate this work, I'm going to introduce two challenges that exist for many of these single photon type of technologies, or even just quantum state photon technologies that enable various implementations of quantum communication and quantum computing. So these types of issues will be the same kind of challenges that exist for companies like Photonic and companies that are involved in this CREATE program. So that make use of photons to do some quantum information processing. Okay, so we are interested in erbium ion emitters, and like some of the defects in silicon emitters, one of the interesting features of these emitters is that they emit in the low loss region of the optical fiber spectrum. So optical fibers are particularly good for transmitting photons in the near infrared region, particularly around 1.5 microns. And you want to use this region because your single photons are precious. You don't want to lose them to losses or scattering. So working in this region is kind of key if you're going to do any sort of single photon or photon number state types of computational or communication schemes. The erbium ion itself has some nice protected long lifetime transitions in this range at 1550. And so this is a very nice source to be using for these types of applications. Now, it does have some challenges. And one of the main challenges, and this is a common challenge to many sort of ion or defect-based emitter application, is that you don't really have a way of isolating deterministically a single emitter. Okay, so here's an example of a work that was done by the Princeton group, and what they've done is they're looking at these erbium emitters in these photonic crystal cavities, And they made several of these cavities, and the erbium emitters are randomly implanted or distributed within this crystal. The issue here being that in some of the cavities, you're going to have just one emitter. In some, you'll have multiple. And the way that they try to get around that was to, for one thing, build multiple cavities to find one that actually had just one emitter. And another thing that they did is they used magnetic field to kind of tune the resonance of the emitter to that of the cavity. Okay, so you can see pretty quickly that this is not a very scalable approach. You can do this for one cavity. You can get some nice anti-bunching measurements that you see on the top right-hand corner there. You can get some lifetime measurements. But the issue is, if you wanted to make many of these emitters at once so that you could do some useful computational communication, that'd be a serious challenge. So how do you isolate just single emitters? And so one thing I want to highlight about this work, though, is that they did use photonic crystal cavities and for erbium. And the nice thing about doing that is that it really enhances the emission rates of these single photon emitters. So erbium, I said, has a long lifetime. It's good. It's kind of a protected state, which is good from a coherence point of view. But it's not good from the point of view of if you want to have a high bit rate communication or have a lot of computational bits, quantum bits happening, then you want to access these photons quickly. And so you need to have relatively fast emission too. So this per cell enhancement that you get from the cavity is kind of necessary and important. We use plasmonics, so metal nanostructures approach to achieve kind of the same thing. Okay, so recognizing that this is a major challenge for quantum sources, particularly those using single emitters, this group took the scheme of doing ion implantation of single ions. So again, not erbium, but from the same sort of family of atoms, the praseodymium ions were implanted into a material, and they trapped them in an ion trap first, and then accelerated them to do implantation. Okay, so already you see this is a super complicated scheme, a real tour de force approach. The issue is that even when you implant a deterministic, well-known number of these emitters, not all of them end up being good emitters. Okay, so if you're close to the surface, they're gonna be quenched out, and so it's not gonna be a good source. So again, we run into the scalability issue here. How are you gonna isolate just single emitters in the place of your photonic circuit that is interesting, or if you're going to add couple to fiber in the place that is interesting for you? And so, you know, one approach you can imagine building on the scheme that they've demonstrated here would be to implant a single emitter and then go back to that spot and see if there's a single emitter there. If there is one, then good, you're fine. If not, then try to implant again and do that repeatedly until you manage to harvest a single emitter. Again, somewhat a cumbersome approach, so we're looking for a more scalable way of doing this. So that's what I want to kind of address in this talk. But that's only one of the things that I want to address in the talk. The other interesting thing that's quantum related and related to our research is looking at these perovskite quantum dots that I've been working on with a bunch of people in this CREATE program. Irina helped us with this work, as well as Maksud Sadamanov in chemistry and electrical engineering. So these perovskite quantum dots are great quantum sources. They have very interesting properties. For one thing, they can be highly coherent. Okay, so here's an example of a paper that's published in Science talking about how really great their quantum coherence properties are. You know, you have emitters that have they're fast emitters, they're bright emitters, but so they give you multiple single photons, well, not one at a time, but multiple of them rapidly, and you get good emission and coherence properties from them, so they can be indistinguishable. The other things that you need for quantum sources. But another interesting feature about them is that they talk to each other very strongly. So if you have these quantum dots next to each other, they're going to basically the dipoles, the optical dipoles of each of these quantum dots is going to talk to each other, and you can get things like superfluorescence. So superfluorescence, for those of you who don't know what that is, it's the property where it's kind of like a laser where you get stimulated or with a laser you have stimulated emission into a single mode. In superfluorescence, all of these dipoles will emit as well into a single mode because they all talk into each other, they all couple into each other, but they do it into a random single mode. So that it kind of happens in a random sort of spontaneous way. So it's kind of a mixture between spontaneous emission because it happens into a random mode, but it's kind of like a laser in that you have multiple emitters simultaneously emitting into that same mode. Okay, so it's a very interesting quantum state of light that's different from a laser and different from spontaneous emission. In this work, a bunch of these quantum dots were built together, stacked together, and then they speak to each other very strongly, and it changes their spectral properties. So the wavelength narrows and shifts. The emission lifetime changes, so they become brighter and faster emitters. And you can look at their coherence properties. All the photons come out at once, so the photon statistics becomes bunched. So the opposite statistics to a single photon source where it's anti-bunched, where you have a G2 of 0 at time 0. So here you have a G2 that's larger than 1. So the chance of getting another photon, a second photon, if you have one photon emitted, is high at time 0 for time 0 delay. Okay, so they have these interesting properties. Also, the light that comes out, it's coherent. It's only coherent with itself, but it's the multiple photons are all in phase and in the same mode. And you get these ringing properties because of that, and a lot of interesting quantum phenomena, quantum optics phenomena associated with these quantum dots. And this is kind of unique, somewhat unique to these perovskite quantum dots. And why? Because they have such strong dipole moments, and their dipole moments are so strong because they have this kind of weak binding. It's kind of for someone who's not in the area, it might not be entirely obvious about why this is, but they have a very large charge or dipole separation, a large dipole moment. Okay. So, but the issue here is that we've looked at perovskite quantum dots. You saw two works that I've shown you here. One was for one single emitters, and the other one was for a collection of multiple emitters. If we want to explore the interaction between these quantum dots, we really want to be able to look at just two emitters. Okay, so that's kind of the thing that we wanted to really understand and explore a little bit in this work, and so that's what I'm going to talk to you about. Okay. To talk about all these things, I need to introduce what we do in our group with it, which is optical tweezers. And our optical tweezers make use of double nanoholes in metal films. Okay, so optical tweezers, the discovery and the invention of optical tweezers was awarded the Nobel Prize in 2018. Half of the prize went to Arthur Ashkin, a quarter went to Donna Strickland of Canada. But his work on optical tweezers really has revolutionized the field of biology, biophysics, because you can use the momentum associated with photons that are scattered away from a focused laser beam to push a particle back to the focus of the beam. And in doing so, you can hold on to that particle and you can learn about the particle itself. And the force of these on the particle is proportional to the polarizability of the particle, which means that as you make your particle smaller, the force gets smaller to the third power of the size of the particle. So that's not great because you can easily trap particles which are about 100 nanometers in size or larger than 100 nanometers up to a micron in size. But if you go down below 100 nanometers, Arthur Ashkin realized that you're going to end up destroying your particles, or you can't really hold on to them long enough. Okay, another issue with using a typical diffraction limited spot is it's huge. You know, you can focus light down to about half a wavelength in a good microscope. But within half a wavelength, if your wavelength is in the infrared, for example, around 1 micron, then that's 500 nanometers. If you're interested in looking at nanoparticles, multiple of these nanoparticles. Here I've shown hemoglobin, which is a tetramer protein, but you know, any sort of quantum dot or lanthanide emitting nanocrystal is kind of the same size. It's tens of nanometers across. And so you're going to have a challenge just isolating just one. Okay, so what do? We use apertures in metal films. The reason we use these apertures is because there you can isolate just a single particle inside the aperture. It has a smaller size because the aperture is nanoscale itself. And the apertures don't allow a lot of light to transmit. And so when you put a particle inside them, they transmit more light because you're making the aperture effectively optically larger. Okay, and this goes as the fourth power of the size of the aperture from Bette's aperture theory. So it tells you that you have this very strong scaling in the amount of light that's transmitted when you have a small aperture, and it's even stronger than the polarizability, the third power that I showed you before. So that means that actually you're going to get more force and more photons acting on the particle that's trapped, and so you get a fission trap in there. Now we did work on this, you know, over a decade ago, coming up on 15 years. I guess it's, yeah, coming up on 15 years now, where we looked at single apertures and we trapped polystyrene spheres there. And I said that when you have a particle inside the aperture, you get more light transmission, and that's exactly what we see. When we focus a laser beam onto the aperture, you see jumps in the light transmission. And with a very small amount of power, less than a milliwatt of power, which is small for an optical tweezer, especially when you're trapping 100 nanometer particles. These are really tiny particles for optical tweezers. We could hold on to it indefinitely, the 100 nanometer particle. But then we managed to push it even further by using these shaped double nanohole apertures. And they just provide some sharpness to the metal to kind of localize the electromagnetic field and make this trapping force even stronger. And we could trap 12 nanometer particles with this. And more than 20 groups worldwide have adopted this and applied this to the analysis of proteins. The first group over here I want to show you, this is Bawendi. He just won the Nobel Prize in chemistry last year for his work on quantum dots. Well, in that work, he used not a double nanohole, but kind of a double nanohole, a double triangle, which is a bow tie aperture, to trap and characterize quantum dots in this type of system. So basically, they applied our approach to the study of quantum dots. But other people have applied this to various things, including proteins and DNA and colloidal particles. So there's been a lot of work in this, and I'm not going to talk about all of that stuff here. But there's more than 20 groups worldwide that are kind of using this aperture approach. Okay, so what do you see in your experiment? I showed you that we see the particles, they're typically floating around in solution. We shine a laser beam. If we look at the transmission through the aperture shown in blue, when a particle gets trapped, all of a sudden you see a large jump. And then the particle moving around in the trap tells us about the size of the particle. And it gives you fluctuations in the intensity of the laser. Tells you about the size of the particle, tells you about the hydrodynamic size, so how it's moving in the liquid. So it tells you a lot of information about the particle itself. And we make these apertures by a bottom-up approach. So we use polystyrene spheres, we drop them on a surface, and then we evaporate gold over, and then we do some sonication to remove the polystyrene spheres. We can also do some plasma etching to precisely control the gap down to 10 nanometers, very in small increments. So we have a very fine control of this aperture or the gap between the cusps of the double nanohole. And as well, because this is a random process, you're going to get single apertures, you're going to get double apertures, you're going to get clusters of 4 or more. But we focus in on the doubles, and we know how to do this by looking at the polarization and optical intensity properties that we see in the microscope. Okay, so now I want to talk about trying to get what I said was the first challenge. How do you isolate just single photon emitters? And for this work, we were working with Frank van Weggel, who's now retired from the University of Victoria. And they make these sodium yttrium fluoride, which is kind of has a low phonon interaction crystal. And inside this crystal, some of the yttriums are replaced with erbium. Okay, and in this case, we looked at very low concentrations, but I'll get more to that in a second. So one interesting thing about these erbium emitters is that they allow for upconversion, which means that with our 980 nanometer trap-in laser, so the wavelength of the laser is 980 nanometers, this erbium ion will absorb multiple photons and then emit light in the green and the blue and the red. So you get multiple emission peaks from this single ion. And it makes it possible to be able to see these emitters, even though they're emitting in the infrared as well, but we can see the visible emission from them. So that's a nice property. So when you trap them, you see clearly this green emission, which is telling you that we've trapped them, even though we use an infrared source. So once we trap them, we try to do this per cell enhancement. So enhance the optical emission from these emitters. And our strategy here was to look at different size apertures. So by tuning the size of the aperture, we could try to tune the resonance of the aperture to match that of the emission wavelength or the excitation wavelength, and thereby achieve more photons out of these emitters. And we were successful in doing this. And in fact, by choosing the correct size of these apertures, in this case, we were using rectangular apertures that we made by a top-down approach called focused ion beam milling. I will get to the other approach later, but in this case, we were tuning the apertures, and we found out that for a certain size of these apertures, you can get 400 times more emission from these erbium-containing nanocrystals. This was exciting to us. Why was it exciting? Well, we realized that we have about 400 of these emitters inside these nanocrystals, and we can observe the nanocrystals, the upconversion from just one nanocrystal. So if we manage to enhance that emission by 400 times, then you realize quickly that actually you could probably observe the emission from just a single emitter. Okay, we've got enough sensitivity in our system to observe a single emitter. And we did a lot of work with Stephen Hughes' group at Queens. He does great nanophotonics theory. I won't go into the details of that, but essentially theoretically it made sense what we were seeing. So what do we do then? We asked Frank's group, Adrian Franken and others in his group to grow us nanocrystals that only contained, on average, 1 or 2 emitters, erbium emitters per nanocrystal. And then we repeatedly trapped these nanocrystals and measured the amounts of emission that we saw from these nanocrystals. And what we observed is discrete levels in the amount of emission. So a nanocrystal that had no erbium emitters would have the lowest emission, well, basically no emission. If it had 1 erbium, it will have a higher emission. 2 erbiums would be more, 3 would be more, 4 would be more, etc. And so doing statistics on this, we can figure out what is the distribution of the number of emitters in each nanocrystal. And it follows a roughly Poissonian distribution. And we did this with 3 different concentrations grown, so very, very dilute concentrations and then slightly higher concentrations. We noticed systematically that the amount of emitters was slightly less than expected from synthesis. So based on stoichiometry, you design your process, your chemistry to give you, let's say, 1 or 2 emitters per nanoparticle, but actually you would end up seeing only 1 emitter per nanoparticle, or slightly less than 1. And the reason that is well understood is because emitters that are close to the surface get quenched. Okay, there's like these dangling bonds at the surface that basically will take the energy away from the emitters. And so you don't really want to, those nanocrystals, although they have single erbiums inside them, will not there will not be any emitters associated with them. So the yield is always slightly less than we expected from the stoichiometry of the chemical process. Okay, so, but that being said, we could trap single emitters, figure out which ones which of these nanocrystals actually had single emitters. And then in the future, we can think of ways in which we can isolate those ones or take them into a reservoir and use them as a resource for quantum information. I'll show you another approach that we did to harvest those single emitters later, but I'll just skip over that for now. Okay, so I mentioned that previously we did rectangular apertures. So the only difference of this work was to look at these double nanoholes that we've been using, and they allow a lot more enhancement in the emission because we can make them much smaller than you can with the FIB. So the focus time beam machine, so we can get, we basically found the separation in the size that gives us the best emission. And with that, we were able to observe even at 1550, which is quite challenging to do in our current setup, but we were able to observe these discrete levels in the emission corresponding to having just one emitter or two emitters or three at the single photon wavelength that we want to use to do sort of quantum information approaches. So that's kind of a good sign. And so now the next step would be to do G2 and HOM measurements on these Hong-U-Mandel measurements. So basically looking at the indistinguishability of the photons that come out on these types of single emitters. Okay, so I mentioned how we are interested in actually, once we've figured out that we have a single emitter, how are we going to isolate it or make sure it stays there. And one approach that Frank's group came up with, Adrian came up with, was to come up with a surface chemistry that could be photocleaved. So we would trap a nanocrystal, and then when we had decided that this is the nanocrystal that we wanted, we would apply some ultraviolet light, and that would cleave off a part of this ligand, and it would expose a thiol, which would then attach to the gold. And so by doing that, we could attach these single nanocrystals to the gold after we had isolated just single ones. So that's a nice way to kind of put it in the nanoaperture, and then we'd go and look in the SEM and make sure that we actually had something attached there after the fact. I should mention that there are other groups that are looking at this kind of approach. I did mention earlier that Bowendy's group was looking at quantum dots, but Jerome Wenger's group in France just recently showed with apertures with the actual antenna inside the aperture, they were able to see from quantum dots anti-bunch-in. So using these as single photon sources is not unique specifically to my group, for sure. Another nice thing of these lanthanides that I don't want to really talk about too much here, but we've been working on this as well, is that by looking at the relative intensities of the different emission wavelengths of conversion, you can measure the local temperature. So from the Boltzmann distribution, you can decide what the temperature is, and in these systems, we can figure out when we're heating up the system by increasing the laser power, we can measure the local temperature at the level of the nanometer scale, which is kind of fun. Okay, so the next part I want to focus on was from a CREATE student. This is Hao Zhang, and he was looking at these perovskite quantum dots, and grown by Maksud's group. And so this, you see the kind of size distribution we're looking at here, and what they look like in a transmission electron microscopy image. So we did the same experiment, essentially. We have an optical tweezer system, which is just a microscope with a 980 nanometer laser. And when we get trapping of these quantum dots, we see a jump in the transmission, and we see emission at 520 nanometers or so. This is the wavelength of these quantum dots. Now, one point that I wanted to make is that actually we don't use a Stokes sort of excitation scheme. We use a 2-photon excitation scheme. So actually, the wavelength that we're exciting with is 980 nanometers. We absorb 2 of those photons and then get emission at 520 nanometers. Okay, so it's 2-photon absorption and then spontaneous emission after that. Okay, so we're not using like a 400 nanometer laser in addition. So that really simplifies our setup a lot. Okay, so we can look at the wavelength of these different nanoparticles, so we can trap multiple of these perovskite quantum dots, and we can see how the wavelength varies. But then we can also look at the signal that we get in from the optical tweezer. You notice that we get these fluctuations after we get trap in. The noise increases. That's not really noise. That's the particle moving around in the trap. It's thermal fluctuations of the particle. And so from the thermal fluctuations, we can figure out the size of the particle because it's related to the Stokes drag. So the hydrodynamic drag is related to the size of the particle, as well as the amount of optical scattering you get is related to the size of the particle. And so from that, we can relate the size of the particle to its quantum confinement. And when you make a particle smaller, then the energy blue shifts. Okay, so then you get shorter wavelengths emitters. So really, the smaller particles have shorter wavelength, and they also have a lower standard deviation in the noise. And so we can characterize both the size and the quantum confinement in this system. Okay, and I won't go through all the theory related to that. That's kind of standard optical tweezer stuff. But you can also figure out the size from not only the amplitude of that noise, but also the time constant of that noise. So what is the characteristic time? If you do an order correlation, what is the characteristic decay time of that noise? That will also tell you information about the size. It's also related to the drag and whatnot. So there's two ways of getting at the same information, or various versions of the same information. And so we can figure out the size of the particles and relate that to the quantum confinement. What was more interesting and somewhat unexpected, not typical for our experiments, usually you just trap one because these particles, they don't like to, if you have a stable colloidal system, kind of by definition, particles are not aggregating, right? But with these perovskite quantum dots, it turns out that we usually saw, another one join in the trap. So you get not just a single jump, but a second jump, where you have two of these quantum dots trapped at the same time. And then we could measure the change in the spectrum, the intensity and the wavelength when we had the second quantum dot join the first one. And there was a systematic shift in the energy of the peak, or the peak energy, when we had the second quantum dot join. And this shift, you can think about it when you have two coupled in a quantum system. If you have coupling, just sort of the simplest version of a coupled quantum oscillator model, then you're going to get a shift. You're going to get basically in phase and out of phase coupling. So you're going to have bonded and anti-bonded type of states. And one of them is going to blue shift and the other one's going to red shift. Assuming that you're in the lower energy state, so basically you're thermalizing into the lower energy state, the shift of the red shifted state is going to be given by this expression over here at the bottom. And so we can fit the observed expression or that expression with the observed actual distribution of shifts that we observed in multiple experiments. And when I say multiple experiments, there really were a lot of different experiments that were done. Individual quantum dots, taking spectra on each one when they trapped one and then two, before and after, and seeing the shift in each of those. Okay, so again, we asked Steve Hughes to help us with the theory because he's an expert not only in photonic crystals and nanophotonic systems, but also with quantum dots and their emission properties. And so the thing that was kind of really puzzling to us in the system was, what was the coupling mechanism? And I don't think this was really well understood. There's a lot of different ways that these quantum dots can couple to each other. They can couple by actually transferring electrons. They can couple by some charge interaction between electrons. There's other, so there could be a tunneling process. There could be a charge related process. There could be resonance energy transfer, which is kind of what we observe here, where you have two dipoles, and in the near field, one emits and it transfers its energy to the other dipole. And if you have that happening coherently, then it will transfer back and forth, and you will get splitting happening. So that's the type of process. When you have degenerate dipoles, that's what you expect to observe. Interestingly enough, for the geometry that we have here, for the type of perovskite quantum dots, it turns out that the resonance energy transfer we found was the strongest process by far, which is a bit surprising, because if you take other types of quantum dots, you wouldn't see this level of split-in or energy shift from this process. And this is because of the type of coupling that these quantum dots have. Their dipole moment is very large. Okay, so it's so large, in fact, that we were hoping to observe some sort of plasmonic enhancement, some enhancement coming from the metal nanostructures here. It turns out that the metal almost doesn't make a difference. So these quantum dots have such large dipole moments already that they talk to each other, but they don't really speak to very much else. And we're not the only people in the world to sort of discuss this effect. People in the solar cell community were interested in using these types of perovskite quantum dots to enhance the absorption of silicon materials. And they found that the quantum dots, again, would not talk to the silicon at all in the absorption. They just spoke to each other. So the impact of putting them on top of a silicon substrate was basically negligible. I mentioned previously that we used 2-photon excitation. To prove this, you would expect that the power of emission would scale as the square of the incident power, because you're absorbing 2 photons. So if you double the intensity, then you expect to have 4 times as much emission, and that is indeed the case. Or you require double the intensity to obtain that. Okay. So that's just a side note there. Okay, so that's essentially all I wanted to speak about with the quantum side of things. I can briefly talk about some of the nanomaterial stuff. Do I have time?
We have it's at 41 past, so but we started a little bit late, maybe another 5, 8 minutes.
Okay, I'll leave some time for some questions. So I just say that this system, this optical tweezer system, we mainly use it to look at proteins. It's interesting for quantum materials. There's other types of materials which are somewhat quantum or interesting for the quantum space, and those are these 2-dimensional materials like HBN, a hexagonal boron nitride, which is kind of like graphene, but it's got boron and nitrogen in its lattice. And so as well as these other types of 2-dimensional materials that we're looking at here. So we looked at these flakes of hexagonal boron nitride. Now these are 2-dimensional materials, so they literally are just like 50 nanometer across, but the thickness is below a nanometer kind of size per layer. And so we trapped these guys in our optical tweezer system, and surprisingly, you see a lot of signal when you trap them. So they give very large signal. But not only that, you see second harmonic generation. So this might not be surprising to many of you who don't work in the field, but for people who work in the field of nonlinear optics, to observe harmonic generation from a single nanocrystal with a CW, so with a low peak power laser, is quite surprising. You would expect to, usually people who do nonlinear optics, they use these intense, ultra-fast lasers. And particularly if you're looking at nano materials, you really have to rely on that huge peak power that you get in a pulse laser, which gives you 1,000 times increase or 10,000 times increase in the local, in the instantaneous intensity to observe these harmonic generation, because the nonlinearities are typically so small. So what this tells you is that the nonlinearity associated with these 2-dimensional materials is huge. Okay, and nonlinear materials are used a lot in quantum optics to give you spontaneous photons, for example. SPDC is one example of a nonlinear process that gives you this advantage. Okay, so just looking at the transmission of the light through the aperture for different types of materials, tungsten disulfide, molybdenum disulfide, and HBN, we see the expected power dependence on the emission, but we also observe that the HBN and tungsten and molybdenum disulfides, they have a lot more harmonic generation, so second harmonic generation, than even a nonlinear optical crystal. So lithium niobate is one of the most popular nonlinear materials. It has a large second order susceptibility. So it is a good nonlinear material, and our collaborators at Simon Fraser University provided us with some samples that they had made, Byron Gates, which are these lithium niobate nanocrystals. Compared to those nanocrystals, these two-dimensional materials have orders of magnitude, even though they're just a single layer, more second harmonic generation. So that was quite surprising and interesting to us. I did want to mention that we do single protein work. It's not really the topic of this talk, but there's other companies that are looking at, like Nanopore and Refine, that have techniques for looking at single proteins. And indeed, we can use our nano tweezers to look at single proteins. And also, we can size the proteins, just like I sized the quantum dots earlier, and figure out, how big is this protein? What are its interaction properties when it interacts with another protein, for example. We can size even the smallest proteins. I have a video here, which I'm not going to show. I'll just skip over that. And with that, I'll just conclude that we're are working on this nanoaperture tweezer technique that we developed 15 years ago, and the double nano hole we developed like 13 years ago. And we're using this to look at single erbium emitters and as a quantum resource. Quantum dots, perovskite quantum dots, as a very strong quantum interacting material. And we're interested in the nonlinear properties of quantum materials as well, which can also be used for quantum information processing. And we're hoping that actually the technique of the tweezer part, well, we're not hoping, we're trying to figure out ways of commercializing this so that even more groups around the world can use it quite easily. And with that, I'll thank you for your attention. And this is an older picture of my group, but they've changed somewhat right now. And Hal's not in this picture. He did most of the work that I talked about today, but I'll just say thank you to them and the funding agencies as well, and I'll take any questions.
Right.
Okay, good.
There is one question that was going back to I'll read the question in full.
Sure.
It's a naive question. So, the dipole due to what species, atom or nucleus? And it was very far back. I think it was in the first third of the talk, when you were talking about the different types of apertures. So you must have said dipole at one point, and I think the questioner was just a bit confused about where that dipole was coming from, whether it was coming from an atom or nucleus or some other source.
Where I was talking about the emitters.
I think so. It was back here, yes.
The perovskites, maybe. So the perovskite is a semiconductor, and so they're excitons. So it's a electron hole pair that are generated, and there's a band gap, and it's the interaction between those two that can emit, and they can recombine and emit a photon. So it's that associated dipole.
If there are any questions that were before that one, I can't see them because I pulled the question.
Okay, I can.
Up. But maybe while people are posting other questions or getting.
There's
it's the person who asked. So, Sarah, if you'd like to unmute yourself, then.
Hi, thanks. I'm the person who asked the original question, so thank you for that. That clears up a lot. But you showed what looked to me like a dense network or a dense of dipoles. So you have quite a dense exciton, lots of excitons?
Yeah, so in this particular study, which was published in Nature, there is a ligand on the outside of these nanocrystals that's separating them. So each one has an isolated dipole inside it. And so if you excite this, then yes, you can have multiple, a dense network of dipoles. Now, the size of the ligand is interesting because if you tune the size, you will actually change the coupling. Not only, and potentially you could allow for things like tunneling. So the dipoles, at some point, it will become a continuous material, right? It's no longer a quantum dot. It becomes just a bulk crystal or a amorphous crystal or something like this. So there's a kind of a tuning that happens if you change the ligand, and some researchers have worked on that. But at the size that we've been looking at, these are essentially isolated dipoles.
Okay. Can I ask a follow-up question?
Oh, sure, yeah.
Or a different question. So I should probably introduce myself. I work at Triumph, and one of the things we do is the implantation of polarized radioisotopes. We're not there yet with rare earths, but certainly to implant heavier species like rare earths is something that is of interest.
Right.
Can you offer a comment about, and so the other thing we can do with our technique is implant at controlled depths within reason. It's not a delta function, but within certain implantation depths between, say, 5 and 200 nanometers. Do you have a comment about, say, tuning the rare earth, or the relevance of depth-controlled rare earth implantation?
Right. Yeah. So that is critical for these rare earths. If you're not looking at the radioactive properties, but just looking at regular visible and near infrared emission, you do want to have the implantation larger than 1 or 2 nanometers, and that's for this quenching property. And I think this is not the case here, where they implanted in this crystal. I forget which crystal they used. Maybe it was yttrium bandionate or something. But yeah, they weren't able to. They were getting quenching, so obviously they didn't have the level of control that would be beneficial in this case. Yeah, it's interesting. Looking at radioactive materials would be interesting too. I'm not sure how comfortable I'd be in my group doing that, but potentially you could apply these exact same approaches to radioactive containing nanocrystals or things like this. And maybe if they had just 1 or 2 radioisotopes, would be fine with that. It wouldn't be.
Let me clarify. The radioactivity is not a problem.
Okay.
And it is typically a single. The technique I'm describing is a variant of NMR, where we implant a beta emitter, spin polarized, and you look at the evolution or the subsequent anisotropic emission of either a positron or electron to give you some sense of the evolution in time of the polarized nucleus.
Okay.
Yeah.
Cool.
And I need to digest your talk, but does that start a conversation for you? Does that have any
For sure.
relevance?
I mean, really, the lanthanide people, they use this magnetic. So I didn't talk about the spin properties at all, but certainly they do have this capability, right? So you can tune the emission by applying a magnetic field, and similarly, you can learn about the magnetic field from the tuning of the emission, so you can. It's kind of complementary to what you're describing there. And as well, we could do similar things on our setup on the nanocrystals. Now, we have done work where also we've looked at the magnetic properties of nanomaterials, like nanomagnetic materials. And by applying a magnetic field and pulling on these particles when they're in the trap and comparing the magnetic force to the optical force, and by that we could characterize the nanomaterial better from its magnetic point of view. We haven't done that for a while, but again, by adding magnetic capability to our tweezer system, you could also influence the spin and probe some of the things they talk about. So yeah, that's a great idea to follow forward.
Okay, thank you.
Thanks.
So as SFU, we have a question. Hi, so is it possible to use a double tweezer setup to look for to generate entangled light?
Entangled light. Yeah.
Yeah, so I think you mentioned at the beginning you were like maybe this is current research, but you were also talking about some Hom experiments. For example, if you look at the perovskite crystals, that one. You mentioned that when they're all correlated, you generate super fluorescence. But I'm wondering, like, let's say we just trap two erbium ions and then they're coupled somehow. And then we do the Hom experiment. Could we see like entangled light maybe that way?
Right. So I think there's a bunch of different approaches that are coming to mind. I can give you an answer, probably not, maybe not the definitive answer on this, but if you trap for the erbiums, they have such weak dipoles, they don't interact with anything. So initially we were thinking if we could pack a really huge density of these guys into a nanocrystal, we could somehow get the superfluorescence from them, but I think that is not really a great approach. There is a group in Sydney, Australia, that's kind of been looking at these nanocrystals that are densely packed, and they've observed that they do get this sort of collective behavior, but I'm not really sure if it's the same thing that we're talking about. So they are kind of resonantly, they looking at resonant excitation. So that would be called resonant Rayleigh scattering. Okay, so that's not exactly the same thing. So getting back to your point, would you be able to see entangled light? I think this perovskite quantum dot system is a good way to do it. Certainly, you might be able to see things like ringing between just two dots if you did a coherent experiment, and you might be doing a pulsed type of laser experiment to see this sort of a pump probe type of experiment to observe coherent ringing or something like this. Already they've observed ringing in these superfluorescent emitters, but which are already kind of entangled in some sense. But if you wanted to have some sort of coherent entanglement between two emitters, that would be one way to do it from just two of these quantum dots. So yeah, I think that's possible if we did get the HOM set up and working with our setup and integrated with the optical tweezer, and we are working towards that. So that is a great possibility. Another thing that we can think about is just looking at SPDC from single trapped two-dimensional materials. So these two-dimensional materials have a very large nonlinear cross-section. So they're very interesting as a potential source of spontaneous parametric down conversion. And one nice thing about using nanomaterials as opposed to bulk crystals is in bulk crystals you have this you're very constrained by phase matching, right? So essentially your photons are emitted in very specific directions based on momentum and energy matching conditions. Like there has to be conservation of both momentum and energy in the crystal at the excitation and the emission wavelengths. And so this kind of gives you really complicated geometries, but at the nanoscale, you kind of relax some of those constraints because essentially you don't have, everything's like point light. So there is no kind of, the uncertainty in, like there's no phase matching along a crystal direction, for example. There's no like propagation that happens. Everything happens in the near field. And so actually it might be a nice way to produce integrated sources that are very densely packed together of entangled photons without having to be constrained by all these phase matching requirements that other approaches require.
Mm-hmm. Okay. Thank you.
No problem.
I have a quick question. So is the emitted light polarized from the trapped nanoparticle?
Yes, it's polarized for two reasons. One is there might be some polarization from the dipole itself, but also the optical tweezer, the apertures have a polarization property. So it forces through the Purcell effect emission into a given polarization state.
Thank you.
So we do see some polarization in the emission. Yeah, we don't really make use of that though. We could do, but not right now. I was just going to show a picture of the apertures. Shows the polarization dependence. There. So this is the graphs on the bottom are showing what happens when we rotate the polarization of the laser, and you see it jumps up and down in the intensity. For a circular aperture, the jumps that you've seen are quite small. That's the first figure here, and the reason it's so small is because essentially you're just probing the polarization and anisotropy of your microscope setup. You know, the beam splitters and the filters and stuff, surfaces that you have in there. But with the double nanohole and those other structures there, you see a much stronger polarization dependence.
Thanks. So we have another question.
Okay.
Okay. Never mind.
Okay. Okay, thank you. Are there any other questions in the chat?
There
Thomas?
are none chat.
Okay.
Since it's 3 p.m., I'll see you after. But
Yeah.
maybe we could all just online give another round of thanks to Reuven for this excellent talk. And unless Bahia has anything to say, I think we'll just see you at the next one.
Yeah, thanks for having me.
Thank you so much.
Thank you, Reuven. Fantastic talk.
And thank you to
Thank you,
everyone
Reuven.
who attended.
Bye-bye.